going after someone telling their own personal story is always quite difficult. I often become really emotional hearing the patient side of it, and I have so many women who I've referred to clinical trials either in my own institutions or across the other side of town, or in a couple of cases interstate. And when they can come back and give me a massive hug and say that I'm really well and I'm going to plan my trip to Europe, well, that does nothing but just fill me with joy as a medical oncologist, but also as a trialist, as a clinical trialist. And it's those stories why we keep going with clinical trials, because if we didn't have the ability to continue to understand what makes a cancer grow and therefore what makes it stop in its tracks and die, well then we may as well not hang up our signs on the door. So yeah, I'm a medical oncologist, but I'm also a cancer genetics specialist, so I have two very different hats, and I really enjoy my two hats because one of them is about largely preventing cancers from occurring, and particularly ovarian cancers. So my interest in familial cancers really came out of hereditary breast and ovarian cancer syndrome, and I still do a little bit of research there, but I've stepped sideways into one of the other hereditary cancer syndromes, hereditary endometrial cancer, and have the amazing opportunity of leading a clinical trial there that we in Australia and New Zealand are going to lead the way in the world. So that's pretty exciting. So enough, let's start. What is research and how does this differ from a clinical trial? I'm going to spend quite a bit of time in this talk just taking a step back and taking us back to the very roots of understanding what cancer is, what a gene is, what makes cancer grow, and hopefully getting to understand what research is, and then where do we get to the clinical trials. So clinical trials are about making available a drug for a illness to make it better, stable, or go away altogether. But there is an extremely long road to drug availability from an idea in a head to actually translating to being able to fill out a script, take it to a pharmacy and receive that drug. And we're going to go through some of those processes today. But if you can see, maybe I'll go here, does this work? Yes. So you can see here, it starts in the design phase and then spends a lot of time going around in a circle here before it might go out into these early phase human trials, and then again there may be some circular motion here before it can get out into the larger clinical trials and before it can actually get out into general population use. So what's cancer research? Cancer research starts with those light bulb moments, and those light bulb moments often come at a clinical meeting. We've just finished the Australia-New Zealand Gynecology-Oncology group meeting. We've been here for 3.5 days now, and often those light bulb moments come when you are in the room together with a room full of clinical trialists and translational researchers and gynecological nurses or people who have an interest in moving things forward. Or in the middle of the night when you're having your hot flushes and you can't sleep and you're waking up and you go, what, this would work. So it starts with a light bulb moment, and you take that idea or theory and you take it into the laboratory and you test out that theory on cell lines, on little single cells. You might move that into potentially a cancer model within a mouse or another animal. But those theories or ideas are about understanding what stimulates cancer cells to grow and what stops the body's normal regulatory systems, like the immune system, from recognising that cancer is an abnormal cell and therefore stopping its growth. So we'll take a step back, and we'll take a step back about understanding what cancer is. And I like to cook. It's just one of my go-tos, relaxation. So I spend a lot of time, doing analogies from the kitchen. So I apologise if you're not into food or the kitchen, but this is just where I start. So cancer is cell regulation gone wrong. So when you do the washing up is a very ordered process. You have a pile of dirty dishes. You have your nice hot soapy water, and you have a draining board, and you have a drying cloth, and then you put them away. And you go from one end to another. And if you get it wrong, it doesn't work. So cell regulation or cell growth is that very ordered process. So what happens in a cancer? Does anyone here in the room remember the film Fantasia or The Sword in the Stone? Any show of hands? No one said, yeah, good. So do you remember the part in the film where Wart in Sword of the Stone or Mickey in Fantasia has to do the washing up, and there are piles and piles of dirty plates to wash up, and they are going through this ordered process. And then someone comes along and casts a magic spell to automate that process and make that process go faster and faster and faster, and in actual fact, it gets out of control and chaos reigns. There are plates going everywhere, there's mops going everywhere, soapy water going everywhere, everyone's slipping over, and it is absolute chaos. That is what cancer is. So it is normal cell division that goes from this one normal, beautiful, healthy cell that should divide into two beautiful, what's called daughter cells, and so on and so on and so on. And if there is an abnormal cell, then the normal process is that the body would recognise that that is an abnormal cell and either repair it or send it off to die, so that it can't go on to make new copies of that abnormal cell. So what happens in cancer is that something goes wrong in this recognition process of an abnormality in a cell, and these abnormal cells are allowed to divide and make new abnormal cells. And the more generations of abnormal cells there are, the less normal they become. They become auto-regulatory. So they have their own growth stimulation. They decide, actually, I'm like Dora the Explorer. I am a cell who likes to go off and explore, so I'm going off to the lungs, or I'm going off to the bones. They take on roles that the original cell would never have dreamed of doing or be able to do. The things that controls the cell process of growing from one into two new cells is largely genetic control. So what's a gene? The genes are like the proteins that control all these cell growth processes. But genes are like a very long recipe. There's a long code. And in order for that gene to work, you need to read from one end of the gene to the other. So, for example, when you're making a cake, you have your ingredients, so your amino acids of the gene that are read in exactly the right way, the recipe, to make a cake. And if everything works well, then you have this beautiful lemon cake with frosted icing at the end. But, so here we go into the science lab. We have our cell, we have our nucleus where all our chromosomes are and our genetic material, and this is our gene being read from one end to another, and if everything works well, you produce a protein at the end of that gene being read. And genes control everything. Genes control eye colour, they control our personalities, they control whether we're sporty sports or whether we like to be just on our own reading a book. They control personality, whether we are party goers or whether we're actually quite insular. They also control hair colour, although we do a pretty good job of overcoming that. But they also control potentially for diseases if things go wrong. So what happens if a gene is abnormal? i.e., in our recipe, if you have a substitution of a P for SR in our recipe, disaster. Things can go very wrong with a single substitution in one gene. Things can become disastrously wrong, just like they can in a recipe, making a cake. So if you think about our normal cell process here of dividing from our beautiful normal healthy cell, this recognition of the abnormal cell and sending it off to die if it can't repair, that protein that we talked about acts here. But if that protein isn't made at all, or if it is not made properly, then this recognition or this death pathway of this abnormal cell doesn't happen, and so cancer cells are allowed to keep going. So cells and cell signalling. You've got our beautiful cell. Inside is our powerhouse here. This is where all this messaging takes place, a bit like a spaceship. On the outside of these cells are what's called receptors, and you can have this signal process from a receptor that goes from the outside of the cell to the inside and creates a whole network of activity within this cell. So in terms of drug development, you can take advantage of this. So if you have something that upregulates this pathway to enable cells to grow, either cancer cell, i.e. cancer cells, and you can block that, then you can block that cancer cell growth pathway. But the pathways, unfortunately, are very complicated and they often feed back on each other. There are multiple things that come in. So just when you think you've got one pathway and block that one pathway, the cancer cell is sneaky and it can come in the back door and activate that same pathway through another way. So if we think about our long road to drug availability, we are back here at our design step. We are thinking about what makes cancer cells grow and what stops them. In a phases of clinical trials, and I know that Tarek touched on this, and in this book there is a really beautiful explanation of the different phases of trials. All that we've talked about so far is in this preclinical stage. It's before we even get to the phase 1, where we think we have a molecule that is going to work against a cancer, but we don't know how much to give. And a phase 1 trial is about finding out a safe dose to give to a human, but that isn't associated with so many side effects that the person isn't able to live and do the things that they want to do, but is just enough to make the cancer cells die. So it's a really tricky balance. A phase 2 is when you've sorted out the dose and then you want to apply that particular drug to tumours that you think it might work in. For example, taking an ovarian cancer and taking you've got this new drug X that targets X, Y, and Z, and you want to take an ovarian cancer population and treat it with a small number of people, around 100 people, to see if it really works in that tumour group. And if it does, then you have to randomise in a phase 3, comparing this new treatment to what is already there. What's the backbone of treatment that is already available? Are we going to do better than what is already there? And finally, in phase 4, if your phase 3 shows that this drug is better and cost-effective and is not associated with greater toxicity, then it is proceeds sometimes to a phase 4 to allow wider availability to people who might have a cancer that is appropriate for that particular drug. So on average, it takes around 10 years and $2.6 billion of research to get one drug from the light bulb moment here into general use. It's a long road. So who runs clinical trials? So we know that a large number of clinical trials are pharmaceutical industry sponsored, and these are often investigator driven. So as a clinician, I can come up with an idea, and I go to a pharmaceutical company who might make a drug that I am interested in trialling in a particular cancer, and I write a protocol, and I do a lot of begging, and I do a lot of writing, and I say, come on, this would be really good, give me some money to do this. That is an investigator-sponsored trial. ANZCOG, the Australia-New Zealand Gynaecology Oncology Group, in combination with the NH and MRC, so the National Health and Medical Research Council Trial Centre, is really the brains trust around a lot of gynecological-based trials, and they coordinate a lot of the clinical trials, but they also provide very valuable links to international groups who are also doing research in what might be appropriate to treat gynecological cancers. But some things to think about. What if you have a really rare type of ovarian cancer, like mucinous ovarian cancer, where the standards of treatments are completely different to the most common sort of epithelial ovarian cancer, high-grade serous ovarian cancer? Well, where do I go to find a trial? And you know, the other thing is that you'll find, and this is about the return of investment on the clinical trials. So you've got the clinical trials aimed at the more common cancers, because they are more likely to generate money back to cover those costs of research and development. And it's easier to recruit sufficient numbers of patients to answer the questions of the clinical trial. Some other things to think about. So my doctor told me my cancer might respond to a type of therapy, but all those trials are currently in different cancer types. So the newer drug therapies, such as targeted therapies or immune checkpoint inhibitors, are now not unique to one cancer type, just like we heard before me. And these are what we call basket trials. So these are enabling the use of a targeted agent to be available for a whole lot of different cancers who have that same potential targetable pathway, so that our rarer tumour groups can get a buy-in, finally, at being able to receive treatments where it may be appropriate. The other thing that is being developed is the concept called tag-on trials. So again, this is allowing those rarer tumours to receive, for example, a drug. I just picked this one, Herceptin, and we'll go back to those mucinous ovarian cancers because we know around 20% of those are amplified for HER2. So can we find some anti-HER2 trials around the world and say, save a place for some mucinous ovarian cancer patients to tag in to those other trials? So this is a new concept that is being worked up. So what about other research? And there's all these other sorts of research. And again, this book goes into fantastic detail about all these other sorts of trials that are available. So clinical trials are essentially about treatment. They're about prevention of cancers, early detection and screening. How can we in fact pick up this in the very earliest stages so that we might do a little simple treatment and then prevent the cancer from occurring in the first place, or treating it in its earliest and highly curable state. Diagnostics, so how can we make these procedures or tests to investigate better? And quality of life and supportive care studies, either during cancer treatment or if you are cured of your cancer but left with a whole lot of symptoms, how can we support you in your surviving years? So we've touched on costs. So some trials cost a lot, but some trials don't cost very much at all, able to make us further forward some understanding. How do we get this money? A huge amount of money for all these clinical trials comes from fundraising. And you guys are the guys that are largely responsible for a huge amount of that money raised for us to be able to investigate and do the things that we need, which is fantastic. not all patients are suitable for a clinical trial, and I'm giving the time, so I'm going to be speeding up here. You need to have the right patients to answer the clinical question, which is why there are inclusion and exclusion criteria, but also in terms of safety. You touched on the fact beautifully that clinical trials patients are followed more closely, and therefore there can be some additional benefits about being looked after in the auspices of a clinical trial. I might just stop there. Take some questions.